All right, welcome to another video. In this one, we're going to give you the background on a simple linear regression, and then show you some output from RStudio and go through the interpretation of that, and then show you in RStudio how we would run a simple linear regression, including creating a visualization of those data. So again, to start, it's a simple linear regression is a statistical method that allows for the summary and study of relationships between two continuous variables. So both your dependent and independent need to be quantitative or numerical variables, ideally both continuous numerical variables. So the regression allows us to test for a relationship between two biological variables, or can one variable help predict the range of another, right? So your independent variable is also known as your X, your predictor, or your explanatory variable, right? So this is the independent variable. Again, both the independent and dependent need to be continuous variables, okay? Your dependent variable, which would be the Y in this case, is often known as your response or outcome variable. Okay? If we look at our statistics roadmap, regression is up here. We need one continuous Y and one continuous X. So again, regression requires two variables, both of which are continuous numerical variables. Again, every statistical analysis comes with a null and alternative statistical hypothesis, right? So the null hypothesis for a simple linear regression right here is that the correlation between variable y and variable x is 0, or there is no correlation relationship between variable y and variable x, or in other words, to say that the slope of a line in a scatter plot with y and x is equal to 0. Okay? The alternative hypothesis, or HA, is that there is a correlation, or the correlation between variable y and variable x is not equal to 0, or that there is a correlation or relationship between variable y and variable x. Okay? So what does this look like? So this is the null hypothesis, right? So here we have blood cholesterol and fat intake. So fat intake would be our independent, blood cholesterol would be our dependent, y and x. The null hypothesis assumes that the slope of this line, a line of best fit through our data, has a slope of 0, or that the slope does not significantly differ from 0. Okay? So that's the null hypothesis. The alternative hypothesis is that the slope of that line is not equal to 0. Okay? And really we're just testing as to whether the slope of the line, the least squares line, significantly differs from 0 or not. That can be positive, it can be negative, or it can be 0, right? We're just testing to see if the slope significantly differs from 0. If it does, in this case, right, there would be a relationship or correlation between X and Y, or fat intake and blood cholesterol. If it does not significantly differ from 0, that's the null hypothesis, there is no relationship between those two variables. Okay? We're not testing for cause and effect, we're testing for a correlation between two continuous variables. So again, the statistical relationship does not equal a functional relationship. We're really just fitting a best fitting line through our data, through our scatter plot, and we do that through the least squares criteria. This is a line that minimizes the sum of squares predicted errors, or the residual error in our data. So the line that is fit to our data minimizes our residual error. And so again, the regression equation is a linear model, right? So y is equal to the intercept plus some slope of x plus some error, right? So beta naught is the y-intercept for the regression line. Beta 1 here is the slope of the regression line, denoting the relationship between y and x. And of course, there's some amount of unexplained error, right? That's the epsilon there, the error. Okay? So again, what does this look like? You've created lines from the y equals mx plus b since high school, right? And so y equals to the intercept plus the slope. We're just adding this little bit of error right? And that error is the residual error. It's the sum of squares between each point, each observed point, and where the line predicts it would be. So in some cases the residual is quite small, or in this case 0, right? In some cases the residuals are larger. We essentially find the difference between predicted y and observed y, right? We find that difference, square them, and the line that is selected is the line that minimizes the sum of squares there. So this is a least squares criteria, so we want to minimize that residual error. Actually, we'll go back here. Again, this beta 1 here, this is the value that we're testing to see if it significantly differs from 0. We're not really testing the intercept. The line minimizes our residual error, so what a regression does is test to see if this value significantly differs from 0. Okay? Like any statistical analysis, there are some assumptions about a simple linear regression. One is that it's linear, right? So it says so right in the name, simple linear regression, right? There's nonlinear regressions. There's lots of different types of regressions. We are teaching you a simple linear regression. So it is assumed that the relationship between x and y is linear, right? This is why when we look at metabolic rate and body size, we log transform them because the raw data are not linear, they're curvilinear. Multivariate normality, this basically just means that both your x and your y variable come from normal distributions, or that your residuals have a normal distribution. No collinearity, meaning no non-independence, so 2 points aren't correlated or connected to each other, right? This is important, and if we don't have the correct experimental design, there's no way of getting around the non-independence. No autocorrelation, that means that residuals are not independent of each other, so it would be like residuals at low values of x are small, while at large values of x are larger. So if there's a pattern to the residuals, that violates the assumption. And then homoscedasticity of variance, or basically normality of the residuals. Okay. So let's look through an example and then do an example in RStudio. So in this case, we're going to use some snake data that I have, and so we have snake body length, which is measured in SVL, or snout to vent length, and that's in centimeters. That will be our independent variable, and then the number of ventral scales, so the number of scales they have along their belly or their underside. Again, that's not technically a continuous, that's discrete, but it's a count. And so we have snout to vent length in centimeters, and ventral scales, or the number of ventral scales for right about 50 snakes, right? We can bring that data into RStudio, again, setting our working directory, reading that data into RStudio by the read.csv file. And then if we look at the structure of that data set, so just naming it snakes, there's 110 observations of 2 variables. So actually this is the larger data set. So SVL is a number, right? And ventral scales is an integer. This integer is, again, numerical and continuous. It's just simply saying that these are all whole numbers rather than having decimals. Okay? Next, we're going to try to visualize our data. So again, 2 continuous variables. The best visualization would be a scatter plot. So here we have number of ventral scales on the y, snout to vent length in centimeters on the x, and all of our plots, our data points here, plotted as open triangles. Okay. And then what we would do is use the LM function in RStudio, the linear model function, to fit a regression line. We can plot that line using AB line or AB line. And here I've done so in green, so we can see that this green line is the regression line between our data. Okay. And then we would summarize that linear model and get an output like this. So here's our call, linear model. The formula is ventral scales tilde SVL. So this is y equals x, right? And then tell it the data frame. Here's our residuals. Here's our coefficient. So the estimate of our intercept is 183. And the slope of the line beta 1 is negative 0.2993. down here is where we look at the test statistics and summary of our regression. So regression, the test statistic is an F statistic. I apologize. So that value is 0.4439 with degrees of freedom 1 and 108, and that's associated with a p-value of 0.507 or 67. Okay? So this is a pretty high p-value, right? So we would fail to reject the null hypothesis, suggesting there is no relationship between snout to vent length and the number of ventral scales that a snake has. Okay? The other thing we look at is the R squared, and typically we look at the adjusted R squared, which helps make sure we're being sort of cautious in our interpretation. What the R squared tells us is how much of the variance is explained by a regression line, and we want this value to be as close to 1 as possible. This is pretty far away, right? Our adjusted R squared is negative, but if we look at the normal R squared, 0.004, so 0.4% of the variance in our data is explained by a regression line. And again, if you look, there's quite a spread of the data. And so the line doesn't really explain a lot of the variance, which fits with the fact that it's not significant, right? Because there's no relationship or correlation between these variables, we wouldn't expect a regression line to actually explain a significant portion of that variance, right? So again, we would report the F statistic, the degrees of freedom, the p-value, and it's common to report our adjusted R square. So that's and then statistically we would fail to reject the null hypothesis and state there is no relationship between the number of ventral scales and a snake's snout to vent length, right? Biologically, that basically means that the size of the snake is not related to how many ventral scales they'll have, right? Okay, so let's look at how to do this in RStudio. Again, I've already downloaded the data and put the data in my directory folder, right, as a CSV file. Again, students tend to struggle here, right? You have to download the file. That file will typically be on Blackboard, and go to your downloads folder or whatever folder you have set as default for your downloads. You then have to move that file from your downloaded folder to the folder that you set as your working directory. In this case, I use RStudio SP24. That's my working directory for this semester. Okay. Again, in a Mac, sometimes when we download a CSV, your Macs will automatically convert it to a numbers file. Then we would have to move it to our folder and then export it in that same folder as a CSV file. Okay, it has to be a CSV file, and it has to be in the folder that you're using as a directory. Otherwise, it doesn't matter if your code's right, it's not going to work. Okay. Again, there's other tutorials on that, so if you're struggling with that, please see those tutorials. So I'm going to set my working directory here. Okay. Then I'm going to import the data. This data is about the number of chirps that a cricket makes in relation to environmental temperature. So I'm going to call that chirps. So here I'm just naming the object. So I'm telling R to create an object named chirps, and that's going to be this CSV file. So read.csv and then cricket_data.csv in quotations. That's the name of the file. Okay, and now I can see that chirps pops up over here in my environment window. Okay. I'm going to just alter this real quick so it's easier to see. Okay. Now if I want to see what that data looks like, I can use view chirps, again with a capital V. That should open up the data file, right? So I have two variables, chirps and temp. Chirps is a number of chirps per minute. Actually, I think it's in seconds, but in the tutorial I'm saying minutes. Not a big deal in this case. And then temperature in degrees Celsius. Okay. If I want to look at the structure of that, I can use str, structure of chirps, right? So I've got 15 observations of two variables. Chirps is numeric, and temp is numeric, right? So I have two numerical variables. One is the independent, and that's temperature, right? And one is the dependent, and that's the number of chirps per second. And so they're both continuous. So again, a regression and a scatter plot are the most appropriate things to do. Okay. So then I'm going to plot, I'm going to create my scatter plot, right? So here's plot y tilde x. Again, chirps is my dependent, so I list that first. Tilde and then x are my independent, that's temperature. And then I tell RStudio what data set to look in. Data equals chirps, right? Comma. And then I have my x-axis label in parentheses, environmental temperatures in degrees C. Y-lab, my y-axis label equals cricket chirps per minute. And actually, let's just change this on the fly, because now that I remember it is in per second. Okay. And then my main axes, or my main title of my graph, again in quotations, cricket chirps per second versus temperature. Okay. And then we have this PCH. Okay. PCH is telling RStudio what type of point to make. Okay. And again, you'll have this in the notes, and there'll be a output on some of the documents. But you can use any value between 0 and 25 in base R. Each number is associated with a different type of point. And so I'll show you how that changes here. But I'm going to start with number 20, and then color equals, and then blue. That's telling RStudio what color I want my points to be. Okay. So let's run this, and we can see that now I spit out this scatter plot. Here's cricket chirps per second, environmental temperature, cricket chirps per second versus temperature, and there's my data points. Okay. Now what happens if I change this PCH value? So I'm going to go from 20 to 1. Boom. Now it's open circles, right? So all the PCH value does, now I'll go to 5, changes the type of point that it is. Okay. Go to 16. Now it's filled in circles. If I don't like the blue, right? Let's say I want my points to be green. I just changed that last thing. There's green. That looks even worse than the blue, right? Let's change it to red. That will stand out a little bit better. Okay. So PCH, that value between 0 and 26, just changes what shape the points are, and then color, or COL equals, just tells RStudio what color to plot those as. Okay. In some cases, we'll ask you to do very specific colors and shapes. In others, it's up to you. Okay. So there's my scatter plot. Now what I need to do is actually do the regression. Okay. And so I'm going to have RStudio create that linear model and fit my least squares regression into the data. And I do that through the LM function here. Okay. So linear model LM. Okay, so I'm going to just call that regline for regression line. Okay, so that's what I'm creating a new object called regline, assigned to LM for linear model, and then chirps, oops, I don't want to do that, chirps tilde temperature. Okay, again, y tilde x, right? So the linear model, y equals x, that's what this is, y tilde x, that's our line equation, and then data equals chirps. When I run this, all you're going to see is that now regline pops up in my environment window. Okay? You don't see output yet. The next thing we can do then is plot that line on our scatter plot so that we can visualize our least squares regression line, and we do that through AB line or AB line. Okay? So that tells us to put a line on our scatter plot, and it uses the object we just created, regline, as the information to plot that line, and then we give that line a color. Okay, I have it as red. Now I'm going to just change it to black so it stands out from the points a little bit better. And again, I run this, and now I can see my scatter plot has a line, right? So here, if I just rerun the scatter plot, right, just a scatter plot. Now the LM, the linear model, runs the regression. That object then can be used to plot the regression line, and I do that with AB line or AB line. Okay. AB line, the object, which is regline, and then the color, right? And now it puts that out on my scatter plot, and now I can save this as an image and use it in my assignments, right? And then finally, I need to actually summarize the regression. I need to see the output of the regression, so we do that via summary and then regline, because that's the actual regression, what we named it, right? So if I run that summary regline here down at the bottom, we see the same things. Here's my formula equals chirps y and temp x. There's my residuals. There's the intercept estimate of 0.4591. Here's the slope estimate of 0.2. Each one of those has a t-value and a p-value. You can ignore those for now, right? This is where we want to look down here, okay? So the F statistic is 29.25 on degrees of freedom 1 and 13. The p-value is 0.00011195, right? And the adjusted R-square is 0.6686, okay? Those are the important pieces of statistical information on this regression, right? So how do we interpret that? We would say that the regression resulted in an F statistic of 29.25 with degrees of freedom 1 and 13 and gave us a p-value of 0.00011195. Since that p-value is less than 0.05, again, 0.05 is just arbitrary, but that's what we go with, right? Then we can have evidence to reject the null hypothesis and say there is support for the alternative hypothesis. This suggests that there is a relationship between the number of cricket chirps per second and the environmental temperature. Okay? Since the slope of that line is positive, we can go a little bit further in our biological interpretation, and we can say that as environmental temperature increases, and now that I'm looking at the actual data, this has got to be Fahrenheit, not Celsius, otherwise it's really, really hot, right? But as temperature increases, the number of chirps per second increases, okay? So there's a positive relationship between the number of chirps per second and the environmental temperature. And our adjusted R squared is 0.6686, which means that roughly 67% of the variance is explained by our regression. Okay? So again, we would interpret both statistically and biologically, and that's about it, right? Thank you.